Okay, so we've been spending some time talking about cellular respiration and why you do it and what you need to do it. So I'm going to talk for a minute about what happens to this metabolic pathway if you don't have oxygen involved. And so I'm going to do a quick review. I'll put a timestamp up here somewhere so that you can just skip ahead if you think you're already good on cellular respiration and you just want to talk about fermentation. So let's do a quick review really quick. So again, this is cellular respiration, and the first part of this process is taking this 6-carbon glucose molecule. I drew it more complicated than this, but it doesn't matter. But you're taking this 6-carbon glucose and you are splitting it up into a couple of different molecules of pyruvate. There are intermediate steps. There's actually a lot more intermediate steps than what it's showing here. Ultimately, what you're going to wind up with is 2-pyruvate. Okay, and what else? You're also going to end up with ATP. You make some energy as part of this process, right? You also make NADH. You are making electron carriers as part of this process. And so what's going to happen is these pyruvate are going to go into the mitochondria. So this whole part of the process, everything sort of beyond this line, is taking place inside the mitochondria. You're going to take this pyruvate, you're going to remove a CO2, and you're going to harvest the electron from that reaction, giving you an NADH. And you're going to continue breaking this glucose down in the citric acid cycle. So this just looks like a fun little circle. There's actually a lot of reactions going on here, but I'm not worried about it. I want you to know conceptually that you take a glucose, you break it into pyruvate, then you take those pyruvate, you put them into the mitochondria and remove a carbon, which is where your first carbon dioxide comes from, and you continue breaking that molecule down in this cycle where you get more carbon dioxide and you get more electrons. You also get a little bit of energy out. So continuing on through this process, so now we are inside of the inner mitochondrial membrane. So the mitochondria has 2 membranes. The mitochondria will have another membrane out here somewhere. And then the cell's outer membrane will be way up there, someplace way up there. So what have we done, right? We took a glucose molecule, we took the energy from those bonds, and we harvested them in the form of these electrons. Now we brought these electrons inside the mitochondria, and we're dropping these electrons off at the electron transport chain. So the electron transport chain is going to transfer, it's going to pass these electrons through these proteins, and passing those electrons through is going to cause these H pluses to get pumped out, and it's going to build up that big charge here in the intermembrane space. But the thing is, those electrons need some place to go at the end after they've been pumped through this electron transport chain and helped us to pump out these H pluses. These electrons need somewhere to go. And so what's going to happen is, it's not emphasized here, but what's going to happen is O2, air that you breathe in, or oxygen that you breathe in, is going to come in and it's going to grab these electrons and it's going to make a water molecule. Okay, remember the whole point of this process is to pump these H pluses out so it can run this enzyme here called ATP synthase, and we're making a bunch of ATP from this energy. But you need somewhere for those electrons to go at the end of the day. The place where those electrons go, if you have enough oxygen, is they bind on, or they become chemically bound to this oxygen. And I said electrons sort of can be thought of as hydrogens in biology, and they make H2O. Okay, but what happens if you don't have oxygen? Or what happens if, let's say, you're like sprinting and you have some oxygen, but you don't have enough oxygen to keep this process running? Well, you're going to build up electrons here, but you're also going to run out of NAD, because these NADHs are going to come in, these FADH2s, these electron carriers are going to come in, and they're not going to have anywhere to give their electrons to. The system is going to get backed up. And so you'll have a bunch of NADHs and you'll have a whole bunch of FADH2s with nowhere to go, with nothing to give their electron to. But you also won't have NAD+. You also won't have FAD because they'll be filled with electrons. And let's go back a second. You'll notice here that a vital part of the process of breaking these things down into pyruvate is having the NADs take this electron. You have to have the NADs come and take this hydrogen off. If you don't have that, the process of breaking down glucose stops. So what do we do? What can we do about this if we don't have enough oxygen? The answer to that is lactic acid fermentation. So here we have glucose going through the process of glycolysis, again, breaking it down into pyruvate. But what's going to happen here is the pyruvate, instead of continuing on through this process, is going to get converted into lactate. You can also think of this as being lactic acid. So normally, remember what happened to our pyruvate, right? We took a carbon off and we sent that into the mitochondria. In this case, we are just making it into lactate because when we make it into lactate, that allows us to regenerate NAD+. It allows us to regenerate this NAD+. So this process cannot go on forever, by the way. This lactate is going to build up in your muscles, and ultimately you're not going to be able to continue running. You're not going to be able to continue lifting weight. You're not going to be able to continue doing anything because lactate, for one thing, it's not an efficient process. For another thing, this stuff is going to build up in your muscles. Okay, so when you are out of breath at the end of a run, at the end of a workout, or whatever, what you're trying to do is you're trying to build up enough oxygen so that you can get rid of this lactate, so that you can reestablish, cellular respiration, so that you can stop doing this fermentation thing. Because again, and this is a test question, by the way. So the point of glycolysis is to start breaking down glucose and to generate ATP, NADH, and pyruvate, which can continue on in the cellular respiration pathway to generate a whole bunch of ATP. That's the whole point of glycolysis. If you don't have any NAD, glycolysis ceases, right? If you don't have those electrons getting taken away at the end of cellular respiration, if you don't have this process happening, you're going to run out of NAD. So in order to deal with that, we convert our pyruvate, instead of sending it to the mitochondria, we convert it to lactate, regenerating these NADs so that we can keep on breaking down glucose. I want you to sort of be able to see the logic here. Okay, so watch that again, slow it down, think about it. But fermentation is a really important process, and it's something that helps, it helps you to be able to do things that you don't have enough oxygen for now. The other kind of fermentation that can take place is alcoholic fermentation. So pyruvic acid, instead of getting converted into acetyl-CoA, is getting converted into this molecule called acetaldehyde. Acetaldehyde. Okay? And then the acetaldehyde is getting converted into ethanol, and we are regenerating our NAD through the process of converting acetaldehyde into ethanol. Okay, this is what's taking place in yeast. Many bacteria will do this, but fermentation is the reason that we have things like beer and aged cheddar and bread, which is like my food pyramid. So fermentation, super important, okay? It allows you to do things that you don't have enough oxygen for, and it allows other organisms to make alcohol and make all kinds of really interesting and tasty byproducts for us. And so before I said it allows other organisms to make things for us, the other organisms don't care. Like yeast don't care about like making beer for you. But it does allow them to survive in anaerobic environments. It allows them to survive where maybe there isn't enough oxygen. And here are some folks making some beer in these little tiny fermenters here. Oh, this is fermentation of grape juice. They're making wine. I'm sorry. So there is also something called anaerobic respiration. It is different than fermentation, even though it sounds kind of similar. They are different processes. So in fermentation, you are stopping at glycolysis. In fermentation, you are stopping at glycolysis. And so you're getting much less energy, right? If we could just do glycolysis, that's all we would do. We wouldn't even need to continue on with the process of respiration, right? So in fermentation, there's no oxygen, so you stop at glycolysis. You stop after you've broken down that glucose molecule. In anaerobic respiration, organisms use other things as their final electron acceptor. So in this case, these algae use, I think they're algae. Oh, they're bacteria, sorry. They use hydrogen, they use sulfur as their final electron acceptor, and it releases this hydrogen sulfide gas. So again, that's an important distinction though. Fermentation ends with glycolysis. Anaerobic respiration is the full cellular respiration. You still get all of that extra energy. You're still doing the Krebs cycle. You're still doing the electron transport chain, but you're using something else as your final electron acceptor, just something that isn't oxygen.